This recording is part 4 of 4 for Chapter 3, the Cellular Level of Organization. Within our bodies, the different somatic cells have different functions. Liver cells have different functions than bone cells, which have different functions than muscle cells. When somatic cells are newly formed, they start growing and they start performing their specialized functions. During this period of their life cycle, which we refer to as the cell cycle, we say that the cell is in a stage known as G0. G0 is when the cell is growing and performing its normal activities, which are very specialized cell functions. For some cells, this may be an indefinite period. If the cell needs to divide because we need more of that type of cell, then the cell will pass through what is known as a restriction checkpoint to enter into a new stage within its life cycle or the cell cycle. That stage is what we refer to as interphase. Interphase is when that cell, which is our original parent cell, is preparing itself so that it has everything it needs to divide into two identical daughter cells, which are capable of sustaining themselves. Interphase is broken down into three portions. The first stage of interphase is referred to as G1. G1 is important because the centrioles, which are going to be responsible in helping direct the movement of our DNA, as you'll remember, they will produce a spindle apparatus that will connect to the kinetochore of the sister chromatids on the chromosomes. Centrioles are going to make a copy of themselves in G1. In G1 of interphase, we also see normal cell functions and growth and duplication of organelles. If we take one parent cell and divide it into two identical daughter cells, each identical daughter cell will have to have enough organelles to support itself. Another thing that occurs in G1 is protein synthesis. So more proteins will be produced in G1. Normally G1 will last approximately 8 hours. After G1, the cell will enter into what is known as the S phase of interphase. The S phase of interphase is important because this is where the DNA is replicated. So a copy of the DNA will be made so that when that parent cell divides, each identical daughter cell has the same DNA. It's got a copy of the original, and that's what allows those newly formed identical daughter cells to start to grow and perform their specialized cell functions. The S phase of interphase not only includes the replication of DNA, but also the creation of more histone proteins, because the DNA is still going to exist in the form of chromatin, and the chromatin is that combination of that double helix wound around the histone proteins. I remember that DNA is replicated in the S phase because S stands for synthesis, and we're copying one of the most important parts of the cell, which is the DNA in the nucleus. From the S phase, which takes anywhere between 6 and 8 hours, the cell moves on to the G2 phase, which is where the centrioles finish their replication and more protein synthesis occurs. When the cell is ready to divide because it has everything it needs to start to split apart and go into two identical daughter cells, there are different portions involved in this split into two identical daughter cells. Cellular division to go from one parent cell into two identical daughter cells, each containing a copy of the DNA, is a process known as mitosis. Mitosis occurs in somatic cells. Not all somatic cells will undergo mitosis. Normally, neurons and muscle cells do not undergo mitosis. Included in mitosis is cytokinesis, which basically means the movement of the cytoplasm. Let's look a little bit more closely into mitosis. Mitosis includes 4 phases: prophase, metaphase, anaphase, and telophase. Cytokinesis begins in anaphase and ends with the end of telophase. Again, mitosis is the division of somatic cells where we take one parent cell and divide the contents into two identical daughter cells. The average time for mitotic completion is approximately 24 hours. In order to create two identical daughter cells, we have to copy the DNA through a process known as DNA replication before the cell divides. As I said before, not all cells will undergo mitosis. That includes red blood cells after they're matured. There are actually other cells that will undergo mitosis to make cells that will form mature red blood cells. But mature red blood cells cannot undergo mitosis because they have no nucleus. They extrude it or kick it out right as they're going into the bloodstream, so they have more room for carrying oxygen and carbon dioxide. Mature red blood cells do not undergo mitosis. Skeletal and heart muscle cells do not undergo mitosis. As I mentioned before, neurons do not undergo mitosis. Some cells will undergo mitosis at a faster rate than others. This includes things like cells inside the small intestines, hair cells, skin cells, bone marrow cells, and cancer cells will undergo mitosis at an accelerated rate. DNA replication is important because we've got to copy the DNA from that original parent cell before we divide into two identical daughter cells. DNA replication includes 3 phases: initiation, where those two complementary strands that have their nucleotides attracted to one another by hydrogen bonds are going to be unwound by the enzyme helicase. From initiation, we move on to elongation, where complementary nitrogenous base pairs are going to be added onto the exposed strands by way of help with a molecule known as DNA polymerase, which brings in the correct nitrogenous bases to be added to this growing strand of the DNA molecule that's being copied. Termination occurs when the 2 original strands are bound to their finished complementary strands. Replication stops and new DNA is formed. Now, you'll see that there's a leading strand and a lagging strand. The sugars on our double helix are actually oriented in different directions. So one goes up in one direction, the other one goes down in the other direction, and there's a 5' orientation with a carbon on one side and a 3' orientation with a carbon on the other side. That's more detail than you need to know. It's because of that difference in orientation of the sugar phosphate backbone that leads to a slightly different process between adding the nitrogenous bases to the leading strand compared to the lagging strand. That is more information than you need for this course, so we're not going to go into that detail. I do want you to remember that copying the DNA in the S phase of interphase has to occur before the cell divides during mitosis. Remember, when a cell undergoes DNA replication, that DNA is still in the form of chromatin, so it looks like cooked spaghetti. That's not organized for cell division. That won't occur until the cell has passed through G2 and enters into mitosis, which helps to prepare the DNA by organizing it. When we look at a chromosome, a single chromosome, when it is condensed, has a much more organized appearance. When a chromosome has been duplicated, the duplicated portion of the chromosome is found as a sister chromatid. So here we have two sister chromatids. We could say this is the original chromosome, and this is the duplicated portion. Therefore, these sister chromatids are joined together at an area of the chromosome, which is called the centromere. The centromere is the central portion of this duplicated chromosome. On each sister chromatid, there is a special molecule called the kinetochore. We saw the kinetochore earlier. The kinetochore is important because that's where the spindle apparatus, which is produced by the centrioles, will attach to the sister chromatids to pull them apart to be distributed into the newly forming identical daughter cells. So we go from chromosome duplicated into sister chromatids, which form this duplicated chromosome that has a central portion known as the centromere, where the sister chromatids are joined together. Each sister chromatid has a special molecule embedded at the area of the centromere known as the kinetochore. The kinetochore is where the spindle apparatus or spindle fibers from the centrioles will attach to help separate the sister chromatids during the division of the DNA. When I was making the recording of this slide, I noticed there were some words that were grossly incorrect, so I corrected them prior to recording this, and I'll go in and correct the other slides that are in Blackboard. Just the wrong word can make a lot of difference. With the cell making a copy of the DNA during the S phase of interphase, remember that it has to continue through G2 prior to entering mitosis. Mitosis is the actual process of cellular division. There's an acronym that many people like to use for mitosis because it includes 4 phases, which are Prophase, Metaphase, Anaphase, and Telophase. This acronym is PMAT. Some people like to include interphase in front of this to remind them that interphase has to occur. Remember that PMAT stands for the actual process of mitosis. During prophase, what occurs is that the chromatin that cooks spaghetti is going to start to organize itself. So the chromatin starts to condense, forming chromosomes. During prophase, the nucleoli that we saw in the nucleus are going to disappear, and the centrioles, which replicated themselves during interphase, it began in G1, ended in G2, the centrioles are going to start producing little spindles. So we see these little mitotic spindles forming from the centrioles. Here's a centriole on one side of the cell. Here's another centriole. If we look closely at this cell under the microscope, you can see kind of a star-shaped formation of one of the centrioles. Really can't see it on the right side, but we can see it on the left. So the centrioles start producing little protein spindle fibers that look like stars around the centriole itself. Those are called asters. And then the longer spindle fibers produced by the centrioles make up the spindle apparatus, which is often referred to as spindle fibers. Remember, when we look at these two different pictures, one being early prophase, the other being late prophase, this is just a snapshot in time. So as we look at early prophase, we see some things. As we look at late prophase, we may see some differing things. It's easy to tell that by late prophase, the centrioles have migrated to opposite ends of the cell. And as the centrioles move apart to opposite ends of the cell, we also notice that the nuclear envelope begins to fragment. That allows the movement of the DNA into two identical daughter cells. Another thing that's going to happen during prophase is that the spindle fibers or spindle apparatus from the centrioles will attach to the kinetochore located in the centromeres of the chromosomes, and that will help direct the chromosomes into a more organized fashion in metaphase, which is the next phase of mitosis. Metaphase means middle, and the sister chromosomes are literally lined up in the middle of the cell during metaphase. In the bottom picture, which is an actual picture of a slide of a cell in metaphase, you can see the spindle apparatus leading to the chromosomes, which are neatly lined up in the middle of the cell. You may also notice the asters on either side formed around the area where the centrioles would be. So the chromosomes are going to be neatly lined up in the center of the cell, in the middle, and that allows for a very organized fashion to divide the DNA. The next portion of mitosis is referred to as anaphase. The prefix "an" means without. The sister chromatids of the chromosomes are going to be pulled apart, so they are literally without each other. The sisters are being pulled apart. And as this occurs, we see that the sister chromatids are forming a little V shape. And here we see that separation at the bottom where that space is starting to separate the sister chromatids so they are without each other. During anaphase, is when we see the beginning of cytokinesis. The word cleave means to cut. A cleavage furrow, a furrow is a depression, may be visible during anaphase. If we look at this cell down here at the bottom, you can see a little bit of a depression right there. And so it may or may not be visible. Late anaphase is the beginning of cytokinesis. Please understand, I'm not concerned with early and late stages of cells and mitosis. It's all a snapshot in time. You should know what occurs during those phases. However, please know that I'm not concerned with the specificity of early versus late. This takes us to telophase. Some people pronounce this telophase, but I've never used a telephone. I've used a telephone. Therefore, I am sticking with my pronunciation. Telophase occurs when the chromosomes have been separated and stop moving. They begin to uncoil and start turning back into chromatin. You can definitely see a cleavage furrow in between the two newly forming daughter cells in telophase. As telophase continues to occur, you might see a newly forming plasma membrane between the two cells. It depends on where they are within that life cycle. It depends on the snapshot that you see at that moment in time. And as telophase progresses, we see that a new nuclear envelope will form around the nucleus. You may see nucleoli start to redevelop, and telophase is the end of mitosis, when we have those two identical daughter cells that have newly formed, and you can see a plasma membrane between those two newly formed cells, then you know that is the end of telophase. Remember, cytokinesis begins in late anaphase or anaphase and ends at the end of telophase. Now we have two newly formed identical daughter cells. As we said before, cytokinesis is the movement of the cytoplasm so that we go from one parent cell and form two identical daughter cells. It marks the end of mitosis and begins with a cleavage furrow that starts in late anaphase and continues all the way to the end of telophase. Here's a little chart of the cell cycle where we can see the cell is at the end of interphase. The cell has grown and it's got the new organelles that it needs. The DNA was replicated in the S phase of interphase, and then it takes us through prophase, metaphase, anaphase, telophase. Cytokinesis, remember, begins in anaphase, ends in telophase, and then we have two new daughter cells, and the cell cycle continues. Mitosis is regulated at several points other than that checkpoint between G0 and G1. These restriction checkpoints are regulated by a special protein called cyclin, which helps control the cell cycle. And these checkpoints act as ways to double check that things are working smoothly in taking a parent cell and turning it into two identical daughter cells. So there are other restriction checkpoints that check different things, like is the environment favorable to create two cells from one? Is there enough space? Is there enough extracellular fluid that would support those cells? That's one additional checkpoint. We also have checkpoints between G2 and mitosis to make sure that all the DNA has been replicated. And again, is the environment favorable? We have a checkpoint in mitosis between the metaphase and anaphase transition to make sure that all the chromosomes have spindle fibers attached to them. So these checkpoints, again, make sure that everything is running smoothly. They may stop the cell from going through mitosis if conditions are not favorable. If there's not enough space to go from one parent cell into two identical daughter cells that are going to grow and take up space, then the cell may be prohibited from going through mitosis. So if the cell fails to be regulated properly, either because of genetic predisposition or environmental factors, that can cause cells to be produced and grow in an unchecked fashion, which is what we refer to as cancer. Cancer just undergoes mitosis, 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 and those cells never get to turn into specialized cells because they're dividing so rapidly. They never differentiate. They never become the special cells that they're supposed to be. That is a process known as dysplasia or abnormal growth. If you've ever heard of different stages of cancer, the stages of cancer are based on the amount of dysplasia that is seen occurring in the cells under the microscope. Cancer cells are very greedy, so they'll just keep dividing, never specializing, but they'll also send out signals to the body so that blood vessels will start coming to them to feed them with oxygen and nutrients, and that process is known as angiogenesis. So again, cancer is a process of unchecked, unregulated mitosis. When we look at cellular differentiation, remember that every somatic cell in your body, with the exception of mature red blood cells that have kicked out their nucleus, that's called extrusion, they have extruded their nucleus and gotten rid of it, all of our somatic cells that have a nucleus are going to undergo cellular differentiation. Our somatic cells all have the same DNA. They've got the same copy of DNA. However, they differentiate because they only read certain genes within that DNA molecule and only produce proteins in accordance with their specific function. That is how cells differentiate, so that they can have different specialized functions. Initially, when an oocyte, which is an egg, and a sperm cell get together, a light is given off. Those two cells forming that one cell form what is known as totipotent stem cells. These totipotent stem cells are the first embryonic cells that will start to divide, creating what is known as a morula. These stem cells have the potential to turn into any kind of cell within our bodies. As these cells divide and divide and divide, they create a structure called a blastocyst. The blastocyst is going to contain cells that are referred to as pluripotent stem cells. These have the potential to still differentiate into any kind of tissue, such as muscle tissue or connective tissue or epithelial tissue or nervous tissue. However, pluripotent stem cells can't differentiate fully enough to support themselves with specialized functions. As pluripotent stem cells divide, they become multipotent cells. Multipotent cells have the potential to differentiate into different cells within a special lineage. So if we look at cells within the nervous system, there are different types of neurons and neuroglial cells. So multipotent cells can differentiate into neurons and astrocytes and pyramidal cells. Oligopotent means that the cells are now more limited and can only become a few different types of cells. Unipotent are the fully specialized cells that we see in our tissues and in our organs that can only reproduce and generate more of themselves. As adults, we actually do have stem cells in our bodies in certain places. However, most of our cells are going to be in the form of unipotent cells. Apoptosis, or programmed cell death, occurs when a cell either reaches the end of its lifespan or it becomes so damaged, aged, or stressed that it can't perform its function as a cell. What happens is the cell begins to shrink and the membrane starts to form these structures that are referred to as blebs, and you can see the membrane kind of coming in and going out, forming these little outcroppings of the cell and its components. That's membrane blebbing. As the membrane starts to bleb, the proteins within the cell are activated to break down the cellular components. And as enzymes break down the nucleus, the cell will start to send signals that will attract macrophages. Macrophages are cells that will eat up the broken apart pieces of the cell as it is undergoing its programmed cell death. So macrophages will come along and basically engulf these pieces to remove them. So as cells age, cell division is needed to replace cells, also to repair wounds. Sometimes we see local changes in terms of cell division, including hyperplasia, which is an accelerated rate of growth in the number of cells. That's a concept you should be aware of. We have hypertrophic cells. Hypertrophy is when the mass increases. So when bodybuilders build muscles, their muscles undergo a process of hypertrophy where the muscle cells actually become larger. Hyperplasia is an increased number of growth of cells. So we have more cells with hyperplasia, we have bigger cells with hypertrophy. Atrophy is a decrease in the mass of cells. And again, going back to the example of a bodybuilder, if a bodybuilder or any athlete stops being active, maybe due to injury, the muscle mass will actually decrease and those cells will shrink. In terms of cellular aging, there are different theories, including wear and tear or mitochondrial theories that actually both blame free radicals. Free hydrogen ions are free radicals. There's a genetic theory that says that mitosis stops because it's genetically determined. As you'll remember, with our chromosomes that have been duplicated, we have two sister chromatids. Here's one sister chromatid. Here's the other sister chromatid. I'm doing it in a similar but somewhat different color just to indicate that this is the copied portion of the chromosome. Remember, they are joined at the centromere. And they have those molecules called kinetochores. Remember, at the ends of the chromosomes are special structures called telomeres. The telomeres help protect the ends of the chromosomes very similarly to the plastic end of your shoelace protecting the end of the shoelace. And every time the cells undergo mitosis, these telomeres shorten. And so most scientists agree that when the telomeres reach a certain length, when they've undergone mitosis so many times, the shortening of the telomeres to a certain degree is what's going to prohibit mitosis. So the telomeres are the most accepted reason for mitosis to end. That ends this recording for Chapter 3.